Hello, my name is Eric Zudak. I'm the founder and CEO of Tolokin Research Innovations. Thank you for the opportunity to speak with you today. Tolokin was founded to address unmet needs in the immunodermatology research area. It is estimated that by 2044, more than half the U.S. population will be in the skin of color category. Many, if not most, inflammatory skin diseases are more common in skin of color than they are in lighter pigmented patients. Now, this coincides with an already significant and growing shortage of dermatologists, which is expected to increase the demand on primary care to provide dermatological treatment and by extension to support clinical research. So these shifting demographics will necessarily have an impact on both standard of care and clinical trials. Now, in either setting, there are three dimensions of accuracy for measuring severity or drug response. First is the quality and applicability of the instrument used to measure. Next is the data that is available for analysis. And third is the skill and experience of the clinical rater. Now, psychometric scales have become standard in dermatology, generally falling into two categories: global assessment on the left and index style on the right. For several reasons, they tend to skew towards lighter complexions in terms of the descriptions and the signs of disease that are measured. Another challenge is that the NIH minimum standards for race are outdated and somewhat unhelpful scientifically. So we have identified the need to abstract ethnicity from color in dermatology, and I would argue for that in all therapeutic areas. But for dermatology, it's really quite simple. Disease looks different in different skin types. Right? The already troubling term race in this context is worse than that. It interferes with science and understanding. All patients in this series of photos identify as African American, but they're certainly not similar in appearance. So why is that important? This word cloud was created from 11 clinician-reported outcomes across various inflammatory skin disease assessments. You can see that erythema is clearly the dominant sign of disease in all of them. This is a nuanced concept, but for the purposes of this discussion, we will leave it here. Erythema means redness, which is a poor description for inflamed skin in at least half of the skin types. So in that case, the primary endpoint is misaligned with the primary complaint of the patients with darker complexions. The spectrum of human skin is too broad to modify all of the scales for all the skin types. So how do we improve? Well, one idea is to validate the existing scales against skin of color images to improve the skill and experience of the raters. It may sound simple, but the process of scale validation has long been labor-intensive, time-consuming, and slow. But using our Tavos platform, scales can be validated quickly and easily using photographs curated for color variation and in controlled categories. This allows raters to gain experience recognizing disease severity in various skin types, and the comparison of responses supports a better understanding and agreement among raters. In addition, the validation exercise will generate invaluable training content for physicians who have not seen the disease in heterogeneous populations. After testing Tavos, we added contrast and brightness sliders, which we think are critical when you're evaluating darker skin, especially in a digital format. So the next question is about the goals of collecting demographic data in clinical research. This recent study divided drug response between light and dark skin, and even with this very broad classification, they concluded that skin color was predictive of the degree of efficacy for the drug dupilumab. This was a part of the inspiration for the detailed patient profile questionnaire in our eCOA platform, TriCOA. It captures specific skin type, making a granular analysis of ethnicity and skin type possible. It also has the potential to provide new information about color and the prevalence and causes of inflammatory skin diseases. Finally, the Lesion Area Mapping Assistant, affectionately known as LAMA, is a simple-to-use image capture and reference guide for the assessment of severity and clinical response in inflammatory skin diseases. LAMA supports the collaboration between raters of different backgrounds in recognizing and assessing disease severity. It also creates a visual archive of disease across skin types with the clinical opinion of a board-certified dermatologist attached to that photo. The goal is to create better consensus and understanding across all sponsors and the research community. Another big idea is to collect demographic data on the clinicians themselves. to further the understanding of the relationship between the demographics of the rater and how they rate severity in different patients. And this is to support shared learning among the physician population. So to summarize, assessment of skin disease deals in percentages and estimates. It is an inexact science. Improving the community interpretation is where we can have an impact. Actionable insight requires recognizing that ethnicity, culture, and color are distinct characteristics that have a distinct impact on scientific inquiry. This is a small part of the work that we're doing, and we're hoping that leveraging the tools we've created will provide clinicians and researchers with new ways of looking at information and collecting it. Thank you.